Hello students, today we will learn about organ and histotypic cultures and tissue engineering. The cell culture are widely used in the laboratories world over for various purposes. In vitro studies with isolated cells are useful for understanding of many cell functions such as respiration, glycolysis, transcription, translation, cell proliferation. Thus, for the study of biology and many functions, the cells grown in conventional and monolayer cultures may be adequate. However, for the study of integrated cellular functions or organ functions, isolated cells will be not found of much use. Now we'll explain in details. Now let's know about cellular interactions in organ functions. There occurs interaction among various cells in vivo, resulting in a cascade of events. These cellular interactions, mostly due to hormonal stimulation, are very important for the expression of their functions. Let's know about the functions examples. Hormonal stimulation of fibroblast is responsible for the release of surfactant by the long alveolar cells. Androgen binding to stromal cells stimulates prostate epithelium. Besides hormones, nutritional factors and xenobiotics also exert stimulatory effects on the cells to function in a coordinated fashion. Now let's know about organotypic models. The cellular interactions that occur in the in vivo system are not possible with isolated cells. The recent developments in the organ and histotypic cultures focus to create in vitro models comparable to the in vivo systems. The purpose of these organotypic models is to retain the original structural and functional interactive relationships of the organ. There are three broad approaches in this direction. First is organ cultures, histotypic culture, second is histotypic culture, and third is organotypic cultures. Let's know about organ cultures. The whole organs or small fragments of the organs that retain the spatial and intrinsic properties are used in culture. Second is histotypic culture. That means the cell lines grown in three-dimensional matrix to high density represent histotypic cultures. Third is organotypic cultures. In this case, the cells from different lineages are put together in the desired ratio and spatial relationships to create a component of an organ in the laboratory. Now, let's know about organ cultures. The use of organ cultures with reference to structural integrity, nutrient and gas exchange, growth and differentiation, along with the advantage and limitations. Let's know about the all the advantages we will discuss later. Now let's know about structural integrity. The isolated cells are individual, while in organic cultures the cells are integrated as a single unit. The cell to cell association and interaction found in the native tissues or organs are retained to large extent. As the structural integrity of the original tissue is preserved, the associated cells can exchange signals through cell adhesion or communications. Now let's know about nutrient and gas exchange. There is no vascular system in the organ culture, and this limits the nutrient supply and gas exchanges of the cells. This happens despite the adequate care taken in the laboratory for the rapid diffusion of nutrients and gases by placing the organ cultures at the interface between the liquid and gaseous phases. As a consequence, some degree of necrosis at the central part of the organ may occur. Some workers prefer to use high oxygen concentration, sometimes even pure oxygen, in the organ cultures. Exposure of cells to high oxygen content is associated with the risk of oxygen included induced toxicity. Example, metabolite exchange is severely affected. Now let's know about growth and differentiation. In general, the growth cultures do not grow except some amount of proliferation that may occur on the outer cell layers. Now let's know about the advantages of organ cultures. It provides a direct means of studying the behavior of an integrated tissue in the laboratory. Understanding of biochemical and molecular functions of an organ or tissue becomes easy. Now let's know about the limitations. Limitations of organ cultures. Organ cultures cannot be propagated, hence for each experiment, there is a need for a fresh organ from a donor. Variations are high and reproducibility is low. Difficult to prepare besides being expensive. Now let's know about techniques of organ cultures. The most important requirement of organ or tissue culture is to place them at such a location so that optimal nutrient and gas exchanges occur. This is mostly achieved by keeping the tissue at gas limited interface of some supports. Let's know about the supports. First is semi-solid gel of agar. Second is clotted plasma. Third is microporous filter. Fourth is lens paper. Fifth is strips of perspex or plexiglass. In recent years, filter well inserts are in use to attain the natural geometry of tissues more easily. Now let's know about procedure for organ culture. The basic technique of organ culture consists of some stages. Let's know about these stages. First is dissection and collection of the organ tissue. Second is reduce the size of the tissue as desired. Preferably to less than 1 mm in thickness. Third is place tissue on a support at the gas medium interface. Fourth is incubate in a humid carbon dioxide incubator. Fifth is change the medium, that means M199 or CMRL 1066 as frequently as desired. Sixth point is the organ culture can be analyzed by histology, autoradiography and immunochemistry. Now let's know about organ culture on stainless steel support grid. Small fragment of tissue can be cultured on a filter laid on top of stainless steel grid. Now let's know about organ culture on filter well inserts. Filter well inserts have become very popular for organ cultures. This is mainly because the cellular interaction, stratification and polarization are better in these culture systems. Further, the recombination of cells to form tissue like densities and access to medium and gas exchange are better. The four different types of filter wells for growing tissues in the form of cell layers. Now we will discuss. First point is growth of cell layer on top of filter. Second is growth of cell layers on matrix on top of filter. Third is cell layers grown on the interactive cell layers placed on the underside of filter. Next point is cell layer grown on the matrix with interactive cell layer on the underside of the filter. Filter well inserts with different materials, that means ceramic, collagen, nitrocellulose are now commercially available for use in culture laboratories. Filter well inserts have been successfully used to develop functionally integrated thyroid epithelium, stratified epidermis, intestinal epithelium and renal epithelium, that means renal epithelium for it is in kidney. Now let's know about histotypic cultures. Growth and propagation of cell lines in three-dimensional matrix to high cell density represent histotypic cultures. The advantage with this culture system is that dispersed monolayer cultures can be used to regenerate tissue-like structure. The common the commonly used technique in histotypic culture use gel and sponge hollow fibers and spheroids. Now let's know about gel and spongy technique. The cells in culture can penetrate gels or sponges, that is gelatin, which provides a matrix for morphogenesis of primitive cells. This approach has been used for the development of mammary epithelium and some tubular and glandular structures. Now let's know about hollow fibers techniques. In recent years, perfusion chambers with a bed of plastic capillary fibers have been developed. The advantage of using hollow fibers in histotypic cultures is that nutrient and gas exchange is more efficient. As the cells attached to capillary fibers grow, there occurs an increase in cell density to form tissue-like structures. Many workers claim that the behavior of high-density cells formed on hollow fibers is comparable to their in vivo behavior. For instance, choriocarcinoma cells grown in hollow fiber cultures release more chorionic gonadotropin than in a conventional monolayer. Hollow fiber culture techniques are regarded as ideal systems for the industrial production of several biological important compounds. Work is progressing in this direction. Now let's know about three-dimensional cultures. Spheroids in histotypic culture. Spheroids represent the cultures of cells usually formed by the reassociation of dissociated cultured cells. It is known for some years that the dissociated embryonic cells resemble to form a specialized structure. The basic principle of using spheroids in histotypic culture is that the cells in heterotypic or homotypic aggregates are capable of sorting out themselves into groups to form tissue-like architecture. The major drawback of spheroids is the limitation in the diffusion and exchange of nutrients and gases. Now let's know about multicellular tumor spheroids. MCTS, multicellular tumor spheroids, provide an in vitro proliferative model for studies on tumor cells. The three-dimensional structure of MCTS allows the experimental studies related to drug therapy, penetration of drugs, resistant to radiation, etc. Further, MCTS have also been used to study several biological processes, regulation of cell proliferation and differentiation, immune responses, cell death, cell invasion, cell gene therapy. The main advantage of three-dimensional cell culture is that they provide a well-defined geometry of cells planar or spherical, which is directly related to the structure and function. It is now well accepted that the MCTS have like the initial avascular stages of solid tumors in vivo. However, beyond a critical size, that is greater than 500 mm, most of the MCTS develop necrosis, that means death of the cells at the center surrounded by the viable cells. Now let's know about technique of MCTS production. Single cell suspension obtained from trypsinized monolayer cells or a disaggregated tumor is inoculated into the medium in magnetic stirrer, flask or roller tubes. As the incubation is carried out for about 3 to 5 days, aggregates of cells representing spheroids are formed. It is observed that spheroid formation is more efficient under that spheroid formation is more efficient under static conditions on stationary and non-adhesive surfaces. For this reason, agar or agarose coated cultured dishes to which cells do not adhere are frequently used to initiate spheroid formation. Once the spheroid are formed, they are transferred to 24 well plates for analysis. Spheroid growth is quantified by measuring their diameters regularly. This can be done by using a microscope eyepiece, micrometers, or an image analysis scanner. Good growth of spheroids is observed when grown in wells. Now let's know about transparent mosaic spheroids. It is now possible to produce spheroids from cells that have been transfected with different genes. Mosaic spheroids are formed by mixing transfected and non-transfected spheroids in the desired proportions. Now let's know about MCTS co-cultures. MCTS can be produced from heterogeneous cells also, forming MCTS co-cultures. This is comparable to heterologous spheroids spheroids consisting of tumor cells in combination with host cells. Let's know about some MCTS co-cultures. First is MCTS and immune cells. Second is MCTS and fibroblast, MCTS and endothelial cells. Heterospheroids with heterotypic cell interactions serve as good models for studying several in vivo processes, example inflammation. MCTS co-cultures are very useful in tissue modeling and tissue engineering. Now let's know about the applications of spheroids or MCTS. Spheroids have a wide range of applications. First point is serve as models for a vascular tumor growth. Second is for the study of gene expression in the three-dimensional configuration of cells. Third is to determine the effect cytotoxic drugs, antibodies, radionucleotides used for therapeutic purposes. Next point is to study certain disease processes, example rheumatoids, arthritis. Next point for the development of gene therapies for several diseases, example cancer. Next point is to evaluate radiation effects on target tissues. Next point for the development of tissues and tissue models. Now let's know about organotypic cultures. Organotypic culture basically involves the combination of cells from different lineages in a determined ratio to create a component of an organ. With the advanced in the organotypic culture techniques, it is now possible to develop certain tissue or tissue models. Skin equivalents have been created by co-culturing dermis with epidermis with interviewing layers of collagen. Models for prostate and breast. Models for control of growth and differentiation of lung. Now let's know about tissue engineering. Tissue engineering refers to the application of the principles of engineering to cell culture for the construction of functional anatomical units. The ultimate purpose of TE means tissue engineering is to supply various body parts for the repair or replacement of damaged tissues or organs. Tissue engineering may be regarded as the backbone of reconstructive surgery. It is possible to supply almost all surgical implants like skin, blood vessels, ligaments, heart valves, joint surfaces, nerves through the developments in tissue engineering. There are two schools of thought while dealing with tissue engineering techniques. Some workers believe that the living cells possess an innate potential of biological regeneration. This implies that when suitable cells are allowed to grow on an appropriate support matrix, the cells proliferate and ultimately result in an organized and functional tissue. This tissue resembles the original tissue in structure and function. This approach is very simple and economical, although the success is limited. Next point is, according to the second school of thought, there are several control processes to produce a new and functional tissue. Thus, tissue regeneration in vivo or tissue production in vitro are very complex. Therefore, tissue engineering is not a simple regeneration of cells, and it requires a comprehensive approach with a thorough understanding of cellular configuration, spatial arrangement, and control process. Tissue engineering is a complicated process. Some fundamentals and basic aspects of tissue engineering with a special reference to the aspect we will now discuss. First point is cell sources and culture. Second is cell orientation. Third is cell support materials. Fourth is design and engineering of tissues. Now let's know about cell sources and culture. Adequate quantities of cells are required for tissue engineering. There are three types of cell sources: autologous, allogenic, and xenogenic. Now first point is autologous cell sources. Let's discuss about this. The cell source is said to be autologous when the patient's own cells are used in tissue engineering. This is a straightforward approach. A piece of desired tissue is taken by biopsy. It may be enzymatically digested or explant cultured, and the cells are grown to the required number. The main advantages of autologous cells in tissue engineering are: first is avoidance of immune complications, second is reduction in the possible transfer of inherent infections, third is next point is let's know about the disadvantages. First disadvantage is it is not always possible to obtain sufficient biopsy material from the patient. Second is disease stage and age of the patient will be limited factors. Now let's know about allogenic cell sources. If the cells are taken from the person other than the patient, the source is said to be allogenic. The advantage of allogenic cell source are: first is obtained in good quantity from a healthy donor. Second is cells can be cultured in a large scale. Third is cost effective with consistent quality. Fourth point is available as and when required by a patient. The major problem of allogenic cell source is the immunological complications that may ultimately lead to graft rejection. The immune responses, however, are variable depending on the type of cells used. For instance, endothelial cells are more immunogenic while fibroblasts and smooth muscle cells are less immunogenic. The age of the donor is another important factor that contributes to immunological complications. Thus, cells from adult donors are highly immunogenic, while fetal or neonatal cells elicit little or no immune response. Now, let's know about xenogenic cell sources. When the cells are taken from different species, the sources are said to be xenogenic. This approach is not in common use due to immunological complications. Now let's know about culture of cells. The methods adopted for culturing of cells required for tissue engineering depend on the type of functions of cells. For most of the cells, the conventional monolayer cultures solve the purpose. The major drawback of monolayer cultures is that the cells may lose their morphology, functions, and proliferative capacity after several generations. Now let's know about genetic alterations of cultured cells for use in tissue engineering. Gene therapy can be successfully employed in tissue engineering. This can be achieved by the transferring the desired genes to the cells in culture. The new genes may increase the production of an existing protein or may synthesize a new protein. Some success has been achieved in this direction. First point is genetically altered fibroblasts can produce transferrin, clotting factor 8 and clotting factor 9. Modified endothelial cells can synthesize tissue plasminogen activator. Next point is genetically engineered keratinocytes can produce transglutaminase 1. This enzyme is lacking in patients suffering from a dermal disorder, lamellar ichthyosis. The altered keratinocytes proved successful when transplanted in animal models of this disease. Now let's know about cell orientation. The orientation of cells with regard to specific shape and spatial arrangement is influenced by some environmental factors. Let's know about the environmental factors. First is substrate or contact guidance. Second is chemical gradient. Third is mechanical cues. First, we will discuss about substrate guidance. The topographical feature of the substrate determine the contact guidance. These features may be in the form of ridges, aligns, fibers, etc. It is possible to use differential attachment to substrate as a means of producing different alignment of cells. In recent years, synthetic polymer substrate collagen fibrils and fibronectin are used as bioresourceable templates for tissue engineering. Next point is chemical gradients. Development of chemical gradients is required for cellular orientation and for the stimulation of cellular functions. Certain growth factors and extracellular macromolecules are capable of creating chemical gradients. Example, vascular endothelial growth factor, in short VEGF, oligosaccharide fragments of hyaluronan, fibronectin, collagen. There are certain practical difficulties in maintaining effective chemical gradients for the cells in three-dimensional cultures. This is particularly the limiting factor when the cells become dense. Now let's know about mechanical cues. The response of the cells to mechanical signals is complex and this may result in any one of the we will discuss the points. Any one of the points, like first point is changes in the cell alignment. Second is deformation of cytoskeleton. Third is altered matrix formation. Fourth is synthesis of regulatory molecules. Example, growth factors, hormones. There are mainly three mechanical cues governing cell population. First is tensional forces. Second is compressional forces. Third is shear forces. Now let's know about cell support materials. The support materials of cells largely determine the nature of adherent cells or cell types and consequently tissue engineering. There are a large number of support materials which may be broadly categorized. First point is traditional abiotic material. Second is bioprosthesis materials. Third is synthetic material. Fourth is natural polymers. And fifth is semi-natural materials. Now let's know about traditional abiotic materials. The traditional abiotic support materials include plastics, ceramics and metals. Traditional abiotic material. The traditional abiotic support materials include plastics, ceramics and metals. These materials cannot be reabsorbed or become biologically integrated into the tissues. Therefore, it is preferable to avoid the traditional material in the tissue engineering. Now let's know about bioprosthesis materials. The natural materials modified to become biologically inert represent bioprosthesis material. They are formed by extensive chemical cross-linking to natural tissues. For the instance, the natural collagen-based connective tissue, example porcine heart values, porcine heart collagen-based connective tissue can be stabilized by treatment with glutaraldehyde. The product formed is not immunogenic than that remains unchanged at the site of transplantation for several years. However, growth of some cells or even connective tissues can occur on bioprosthesis material. The design and fabrication of these materials is done in such a way that their functions are not affected by the surrounding host tissues. Now, let's know about synthetic materials. A wide range of synthetic bioresorbable polymers are available as support materials. The most commonly used polymers in tissue engineering are PGA, polyglycolic acid, polylactic acid, that is PLA, copolymer, that is PLGA, polylactic glycolic acid. The composition and dimension of these polymers can be so adjusted to make them stable in vivo, besides supporting in vivo cell growth. There are certain advantages in using synthetic polymers. Production is easy and relatively cheap. Composition of polymers is reproducible even in large scale production. There are, however, some disadvantages also. First disadvantage is compatibility with cells is not good as natural polymers. On degradation, they may form some products which cause undesirable cellular effects.